having me here to talk about what we've been working in the Philippines. This project is a joint program between Manila Economic Cooperative and the Taiwan Economic Cooperative with National Chongqing University in Taichung, Taiwan. And we are just started off a few months ago, and we are just finishing up our preliminary data from year one. It's a 3-year project. I'm glad that the previous speaker already outlined the disease, and we are all familiar that this has been a very significant vector-borne disease of global importance. And I'm not very happy to inform everyone that the first outbreak was in Metro Manila in 1954 in the ASEAN region. And major epidemics has been going on, reported yearly and all over, beginning off in 54, 66, 83, 98, and 2001 in Manila. And in the Philippines, it's actually one of the top 10 causes of morbidity in the country. And in the previous years, there were high rates even in rural areas, and has been showing a lot of cases occurring into 790 deaths with the case fatality rate of 0.59%. And over the years, we've seen observations of the disease progressing in number. And sometimes we have looked at several factors such as vector, and we all know that the changes in the patterns of our climate actually affects the behavior of the vectors, which allow us to have some information that will let us communicate between the environment, the vector, the host, and even the virus. And majority of studies has been done already on some of the effects of weather disturbances in relation to tropical diseases. And we want to dig deeper into the area. If you look at the slides, this is actually a composite of cases we've seen in the Philippines way back in the early 60s. And those yellow brackets are actually some of what you call the El Nino phenomenon. And we've seen that per every event there is a peak in the case, but the one that we have actually observed is that during the later part of the years, it's not just the relationship of the events, but the frequency of the occurrences of the weather oscillations. And since then, we've never seen already the peak and the trough of the disease, but we're actually seeing further and further increase of total number of cases found in the Philippines. In the Philippines, we have all the 4 serotypes, but I'm sure most of you have heard about that the 5th serotype has been identified and is currently being investigated and evaluated. So basically, we have 4 serotypes, and the most common is Dengue Serotype 3. Similar to what was observed in Singapore, despite the increasing number of cases, we've had also a significant drop in the mortality rates in terms of case fatality rates over the year. And I would agree that this may be related to early recognition and higher awareness of clinicians, or is it because of the changing patterns of the virulence of the virus? And that's what we're trying to figure out, or it's actually a combination of everything. In the Philippines, these are our gaps in terms of dengue programs, in terms of diagnosing and treating and preventing the disease. And certain programs are actually in place in the country, but still there's too lack in some of the areas that most of us in the global community are wishing to have one, like treatment for active diseases like the presence of antiviral drugs or an effective vaccine, and we're still laboring on that at the moment. My institution, the National Institutes of Molecular Biology, are preparing the genomic sequence data with clinical outcomes over the last several years and linking its genomic variations with vector migration, environmental perturbation, and climate change data. We're combining everything from the country and from Taiwan, and we will co-validate our findings in year 3. And these are the matrix that we're looking at, from the dengue virus themselves to the genomics, the enzootic mixing, some of the mutations, and the variability in the virulence of the virus, from rural to urban development, and the patterns of spread within the country for both sides. So basically, we wanted to have the evolutionary data of the virus within the country and to correlate that with what is actually available out there outside the Philippines, and the determination of the phylogenetic change in relation to the incidence density, clustering, outbreak intensity during epidemics or outbreaks it has. But ultimately, at the end of our three-year program, both of our collaborators from Taiwan will develop a global web-based interactive dengue outbreak, pattern of spread and severity mathematical prediction model for the prediction of the epidemic potential, combining factors coming in from environmental data. That, for example, in the Philippines, we have weather data as early as 1954 from the first epidemic, and we will put all of those information, since all of them are already electronically available, and we just need to use that information from our epidemiological data from the Department of Health, from our evolutionary data from the virus, and from the data from the vector migration within the country in relation to the storms. I'm sure you all know that our country is the favorite pathway for storms. And we nearly complete the entire alphabet of names last year. We ended up with letter Y. A few more storms, it will be Z already. So now we have our first, it's called, it starts with A. So it's how we do it. So we ended with letter Y with the Yolanda and basically these events actually perturbs the entire system. And these contributory are the frequency, location, duration. So these are the linear and non-linear information that you want to put together. Where are we now? We are just starting. We are at year 1, and we are looking at the metagenomics profile for both mosquitoes and humans. And we are actually co-collecting with another group who are experts in vector migration to put the vector density profile in relation to the layers. Ultimately, the climate health time series will be of several layers being put together at year 3, and hopefully we'll be able to give you a much better perspective a few years from now. So, just for the information of everybody, there has been stable genotypic predominance in the country. Over the last several years, from the first published genomic sequence, serotypes 1, 2, and 4 has been the same. The only problem was when we were looking at the serotype 3, which is the one that's very common in the Philippines, there was a shift and a different genotypes variation is actually coming out that doesn't match with the rest. And this actually been just recently isolated after we have several changes in the storm events over the last 5 years.
Can you hear me?
I can hear you.
Please ask the mute mic.
Oh.
Please just ask the mute mic.
Can you hear me? Everybody's silent?
Yes, they can hear.
They can hear.
Just go on.
All right. So basically, that's the area. But the proposed ultimate end product is to build a dengue disease detection system in the form of a mathematical model which incorporates all the possible identified factors influencing the occurrence of dengue cases. These are, but are not limited to, environmental factor, rural or urban, population density, vector, species migration and behavior, viral metagenomics, strain, pathogenicity, and genetic profile, including climatic behavioral pattern. And I wish we will be able to provide you this information once we've completed all our data collection. Right now, we are still in the process of data mining from secondary data that is already available and from genomics data that we've been collecting over the last 6 years. And that's my 15-minute talk. Thank you so much for your attention.